So thank you, everyone, and welcome to the Precision Medicine Data and Discovery for Improved Health and Therapy session. We have a long session today, so let's get started quickly. The session chairs this year are Bruce Arano, Stephen Brenner, Sean Mooney, and myself, Alex Morgan. We had 36 submissions, which is quite a lot. Each paper, there was one paper we only got 2 reviews for, but every other paper got at least 3 and sometimes up to 6 reviews. So we really are indebted to all our reviewers, many of whom are here in the audience, and really is impossible, as Larry said, without people proposing sessions, but also without all the reviewers. We were able to offer 12 papers speaking slots, and there are actually 15 papers in the proceedings, so I hope you can take a look. There are going to be numerous posters related to precision medicine right next door, as Larry mentioned, and there'll be lunch served, so you definitely want to check that out. Then we're going to have a discussion section after that. Josh Denny will be speaking from Vanderbilt, will be speaking a little bit about the Precision Medicine Initiative, which obviously is relevant to the topic of the session. And then we're going to try to collect, if the Twitterati in the audience are willing to tweet either to the PSB hashtag or tweet directly to myself. We'll try to collect comments, and we can put those together and find for food for discussion. So hopefully in one slide in under a minute, I'm going to define and explain what precision medicine is, as well as outline all the major challenges in the field. So historically, we've had the art of medicine, which has been about human judgment. So this is the sort of definition of professionalism and medicine as a caregiver and provider, using human judgment and their own expertise. That evolved in the latter part of the 20th century into what was called evidence-based medicine, and this is the rise of algorithms designed to be more, hopefully, better than human judgment on average. And now we're trying to reach precision medicine, and these are algorithms better than human judgment per individual patient. And of course, the corollary of that is it begs the question of how we actually know and are confident and believe that we're actually doing better per individual patient. And there's going to be a session on Friday about reproducible results. We'll dig out into greater depth. But to take a very high-level view of what I think are the major challenges in the field, Bob Nussbaum quoted William Bossert yesterday and said mathematical models are typically either too good to be true or too good to be true. And I think that's the sort of summary of the challenge that we're trying to address today. Mathematical models are typically either too good to be true or too true to be good. So, sorry about that. Let's get started with the science.
So I'm Stephen Brenner, and it's my tremendous pleasure today to be able to introduce my colleague, Jennifer Doudna, as the invited speaker for both this session and also for the regulatory RNA session. The regulatory RNA session, just so you're aware, will be held tomorrow at 11:45 a.m. One logistical note, in order to maximize the time for Jennifer's presentation, that we'll be holding all questions until the discussion session this afternoon in this room at 2:30 p.m. Now, one could readily argue that at this point, Jennifer needs no introduction, having been profiled in the New York Times, the New Yorker, and many other places. Jennifer, you may be either pleased or horrified to know that my mom is really excited and proud of me for the fact that I know you, such is her renown within the popular field. So Jennifer grew up on this island in Hilo before going on to Pomona College. She went on to complete her doctoral work with Jack Stosek at Harvard and with Tom Check as a postdoc at University of Colorado Boulder, where she undertook pioneering work in engineering and characterizing ribozymes. And the immediate impact of this work was such that actually when I was an undergraduate at Harvard, just a year or two after the papers were published, this work was actually taught in my undergraduate organic chemistry class. Continuing as faculty at Yale and at Berkeley, she's taken on rigorous approaches to understand the mechanistic basis of RNA regulation. Of particular interest to me has been her work exploring irises and how they can regulate translation, as well as her studies of the RNA interference pathways. And she's garnered numerous awards for these, and I won't begin to list the number of awards she's received, because that would take all the time that we have. A decade ago or so at Berkeley, I started collaborating and interacting with a colleague, Jill Banfield, who was studying a really fascinating community of microbes at a Superfund site called Iron Mountain, which has a drainage that comes through it, which has a pH below pH 1. And so in her metagenomic studies, Jill Banfield became interested in a curious set of repeats that she was seeing in these genomes that appeared to be a record of viral exposures that these microbes had seen, and ultimately were recognized to be part of a microbial immune system. And so I was delighted when Jill Banfield told me that Jennifer Doudna would be joining this project to try to understand this system called CRISPR. And Jennifer did, of course, have a major role in helping elucidate how this system works. And as all the world knows, it also led to discovery of one of the most important tools for manipulating biology, as she will discuss today. So please join me in welcoming Jennifer Doudna.
Great. Well, good morning, everyone. Thank you so much for that generous introduction. It's a really great pleasure for me to be here. As Stephen mentioned, I'm a Hilo girl, so I grew up on the other side of this island, the rainy side. So it's a great pleasure to always, for me to come back to this island. It feels a little bit like coming back home. And this is really where I got my start in science in terms of thinking about the nature of things and my curiosity about the natural world, which has always been, for me, the kind of the underlying driving force for what I've done in my career. So I'm delighted to be here because I really have been on an exciting ride myself over the last few years getting involved in precision medicine. It's not a field or an area that I had any involvement in until the work that I'll tell you about today kind of took us naturally in that direction. And I wanted to start off by just kind of connecting to some of the other themes that we've heard at this meeting so far with the idea that, precision medicine, and really I think what all of us are interested in terms of connecting genotype to phenotype ultimately, comes down to, can we really get information at a gene or even a base pair specific level in the genome that tells us what the function of particular coded information in the genome is doing in terms of phenotype and ultimately in terms of health and disease. And so when I was a graduate student back in the 80s, one of the things that had come to the fore at that time was the recognition, and this was of course before any whole genome sequence was available. We didn't have even the first bacterial genome until 1995. But in the late 1980s, people already appreciated that if you had a way to make very precise changes in the DNA of cells at the level of even a specific gene, but even better if you could make it at the level of a particular base pair, that this would be a powerful technology. And sort of analogous to what I'm showing here in thinking about being able to change a particular word or even letter in the genetic code of a cell to affect a change. And so imagining a technology for that, many scientists had approached this by understanding and going about the work of determining how cells actually make changes in the DNA on their own. And a lot of that work plugged into the natural repair pathways in cells and the appreciation that cells have natural ways of fixing double-stranded DNA breaks to introduce change. So basically a technology for precision change to genomes from our perspective, my own lab's involvement in this really came about not through a focused effort to discover a way to do this, but actually through a curiosity-driven research project that began, as Stephen mentioned, with our work on a bacterial immune system called CRISPR. And so what I'm going to do today is I'm really going to I want to tell you sort of three aspects of this work that we've been involved in over the last several years. I want to first talk a little bit about the work that led to the understanding of the CRISPR pathway at the level where it could be harnessed as a genome editing technology. And then I want to tell you a little bit about how that mechanism, the mechanism of this actually works, and how understanding that mechanism can provide very useful information for making it better and making it into a tool that is very applicable in various different cell types, but also for other kinds of applications beyond making covalent changes to the genome. And finally, I want to say a little bit about what I've been involved in very heavily, extensively, especially over the last year in thinking about ethical and societal questions that come up when we realize that we have in our hands now a very powerful technology for changing the DNA of cells and how we can, as a community, approach this in a responsible way. So for us, really, this work began, as Stephen mentioned, with a phone call that I got from Jill Banfield at Berkeley about 10 years ago, back in, I think it was around 2005 or so, when she called me up and she said that they were looking through the metagenomic sequencing data for a number of the organisms that they were isolating from interesting environments in the wild, these organisms that can grow at pH 1, and of course these are typically biofilms. These are bugs that are not culturable. They have a very interesting community life with their neighbors and also with viruses that infect them. And one of the things that had emerged from her own research, as well as at that time just a few other labs around the world, was the observation that many bacteria have a repetitive sequence in the genome, sometimes more than one, that occur in clusters. And so these had come to be called clusters of regularly interspaced short palindromic repeats, or CRISPRs. And these consist of typically 40 or so base pair sequences in the DNA that are recurrent over and over in a pattern. And these are separated by sequences of about the same length, typically 30 to 40 base pairs, that were initially kind of mysterious. They were called spacers, but in 3 papers published in 2005 by 3 different bioinformatics groups, these sequences were found to occasionally at least correspond to sequences found in viruses or plasmids. And this was sort of the very first clue that these might be signatures of some kind of bacterial immune system, an adaptive way that bugs could acquire sequences from phage or other kinds of foreign DNA elements that get into the cell, create a permanent record of that interaction or infection in the form of an inserted sequence in the CRISPR locus, and then potentially allow the cells to use that genetic information to protect the genome in some way from future infection. Totally a hypothesis at the time. There was no experimental data, but another interesting clue was that next door to these CRISPR sequences were very typically CRISPR-associated or Cas genes that co-occurred with the CRISPR loci. So it very much had the look of some kind of a system or pathway in these cells. And so Jill contacted me actually because, as Stephen mentioned, we've had a longstanding interest in my lab in the function of RNA molecules in controlling the expression of genetic information. And one idea was that perhaps these bits of viral DNA incorporated into the genome were in fact employed at the level of RNA molecules that could go around and survey the cell for DNA sequences that had a match to these sequences here in the form of RNA. And so what emerged over the next few years, both from work from our own lab, but from several other labs, and very importantly, I want to mention too, that really made key contributions early on in this field. One was the work going on at Danisco, a yogurt company. So Rodolphe Barango and his colleague Philippe Horvath were working on this CRISPR system in the types of bacteria that are used to culture yogurt and cheese, and they figured out from genetic experiments that in fact these CRISPR systems do form a bacterial adaptive immune system. And then work that was going on in the lab of Jean van der Oost in the Netherlands showed that these systems actually function at the level of RNA protein complexes that assemble. And so let me just walk you through this briefly. And so what we know now is that in bacteria that have one or more CRISPR systems, they can acquire sequences from foreign DNA. They have a very interesting integrase that integrates new foreign DNA into the CRISPR locus in the genome of the host. And I won't tell you about it today, but we've done a lot of work in the lab also to understand that part of the pathway. And then once those sequences are integrated at the level of DNA, they're transcribed into RNA molecules. Very typically, these are produced initially as long transcripts that are subsequently processed into shorter RNAs. And importantly, each of those produced RNAs has one of the integrated sequences incorporated into the molecule, so that this sequence at the level of RNA is then incorporated into a complex with proteins. These are proteins that are encoded in the CRISPR-associated or Cas genes to form RNA protein interference complexes. And they use the information in the RNA to base pair with matching DNA sequences and allow one or more of these associated proteins to cleave the targeted DNA. So it's a very nice way that bacteria can actually acquire immunity to foreign DNA elements and target them for destruction in the cell. And so in my lab, we actually started working on this sort of central part of the pathway, namely how RNAs are produced, how they're processed, and then how they assemble into protein RNA targeting complexes that can interact and cleave DNA. And it was really through that effort that I ended up starting a collaboration with Emmanuel Charpentier's lab and working together on the function of a particular Cas protein called Cas9 that's found in some types of CRISPR systems and was known at the time genetically to be the only protein in that type of CRISPR system that it was essential to protect cells from a viral infection. And so working together, we've figured out that Cas9, which is shown here in this cartoon, is actually a fascinating protein that has the ability to interact with double-stranded DNA sequences, unwind them, and generate a blunt double-stranded break through 2 active sites in the protein. And importantly, this is a programmable protein in the sense that it binds to a CRISPR RNA molecule shown right here, which contains a 20 nucleotide sequence at the end that can base pair with DNA and trigger this unwinding reaction when it's bound to DNA in the context of the Cas9 protein. And a couple of other things that we figured out from our biochemical experiments. First of all, this system requires a second RNA called tracer, which is shown here in red, that makes an interaction with the end of the CRISPR transcript and forms a structure that binds to the Cas9 protein. So this part of the RNA is essential for assembly of the RNA protein complex. And the other thing I want to point out is that this cleavage reaction requires not only a 20 base pair match to the CRISPR transcript sequence, this sequence right here, but it also requires the site in the DNA to be adjacent to a small motif called PAM, which for this particular Cas9 protein is a GG dinucleotide motif. And so once we had this mechanistic insight into the natural function of the protein, Martin Jinek, a postdoc in my lab who was doing this research, figured out that he could actually simplify the system compared to what nature has done by linking together the 2 RNAs, linking together the CRISPR sequence and part of the tracer molecule to form a single guide form of the RNA that contained both the targeting information and the Cas9 binding information in one molecule. And so this created a 2-component system, a single protein and a single transcript, in which the targeting specificity of the protein could be changed by simply altering the 20 nucleotide sequence right here in the RNA. And just to show you sort of the very, very simple but sort of important experiment that Martin did to test this idea, he basically took a plasmid DNA, designed 5 different single guide forms of the RNA that could interact with a DNA sequence adjacent to this little a GG dinucleotide motif on either strand of this plasmid. And then we programmed the Cas9 protein with one of these single guide RNAs together with a second restriction enzyme that cuts about 60 base pairs upstream. And when we analyzed the doubly digested plasmid products after that incubation, we found that each of the little pieces of DNA released in this double digest corresponded exactly in size to the site that should have been cut based on the targeting specificity of each of these single guide versions of the RNA. So this was, for us, I think, really the moment when we knew that we had a system that was simple to reprogram and could potentially be used as a genome editing technology. And let me show you briefly a little bit more about how this works. So this is actually a 3D printed model of the Cas9 protein and its guide RNA bound to a double-stranded DNA. It's a model that's based on a crystallographic structure that was solved by Martin Jinek's lab last year. And this, what you're seeing here is the Cas9 protein in white. And you can see that this protein really has a channel down the center where it grips the RNA in orange. And when it finds a sequence in a double-stranded DNA molecule that matches the sequence in the guide portion of the RNA, the DNA opens up and allows the protein to interact with this nucleic acid through an RNA-DNA hybrid. So you can see the orange-blue double helical RNA-DNA hybridization in the center of the protein. And then the 2 active sites in the enzyme are able to make a clean double-stranded break in the DNA in a very targeted way. So why did we think about this in terms of, what made us think about this in terms of genome editing? And this sort of gets back to what I mentioned at the very beginning with a lot of research that has been going on really for the last several decades to understand how cells handle double-stranded breaks in DNA and how scientists might be able to harness that natural ability for genome editing. And so we know from the work of Maria Jason and many others that eukaryotic cells, plants or animals, eukaryotic cells experience a double-stranded break to their genomic DNA, the cells have machinery to repair those breaks, typically through either non-homologous end joining, in which the ends are ligated back together with a small disruption in the DNA sequence sometimes, or a second pathway here called homology-directed repair, in which a donor DNA molecule can be recombined into the DNA at the site of the double-stranded break if there is some homology between this donor DNA sequence and the sequence of the DNA flanking the site of the break. And so through this pathway, there can actually be incorporation of new genetic information at the site of the break. And so appreciating these pathways, there had been numerous, I think, very clever, interesting ways of triggering double-stranded breaks in a site-specific fashion, beginning with Peter Dervin's work back in the 80s and 90s using chemical approaches to do this. Many people thinking initially about using DNA oligonucleotides to target sequences for cleavage using small molecules. And then eventually working towards using proteins to do this, zinc finger nucleases, talons, homing endonucleases. These are all systems where the protein can actually be programmed through engineering to have a particular DNA binding ability, and then by linking those proteins to DNA cleaving domains, they could be used to create very specific double-strand cleaving, DNA cleaving enzymes. And so I think you all know that these can be very effective for genome engineering. The challenge in terms of adoption as a widespread technology is that they require significant protein engineering for every experiment. And so I think an important advantage of the Cas9 system is that this is a single protein that can be reprogrammed by a single guide RNA to have essentially any targeting specificity that is desired for a particular experiment based on the characteristics that I described. So let me show you a movie that just kind of illustrates the way we imagine this might actually work in a eukaryotic cell, and also explains what some of the questions are that we still really have that we'd love to understand better about the system. So this is a movie that was created by Janet Iwasa at the University of Utah for us. And it basically shows zooming into a eukaryotic cell. So we're going inside the nucleus. You know that in the nucleus, the DNA is packaged into chromatin. And so this is a bacterial system that has to somehow deal with highly packaged DNA, search through the entire genome to find, in principle, a single site in the DNA that has a sequence that matches the sequence of the 20 nucleotides in the guide portion of the RNA. And when that occurs, then this protein RNA complex binds to the DNA. It forms the structure that I showed you in that model, creates a double-stranded break in the DNA, and then has to release this to the repair machinery of the cell to allow this double-stranded break to be resealed, in this example, by incorporation of new information at the site of the break. And remarkably, this bacterial system seems to work in virtually every cell type that people have tested. Many different kinds of plants, fungi, bacteria, of course, and also mammalian systems, including human cells. So in my own lab, we've been very keen to continue to work on the basic mechanism of this, to really understand how this works. And let me tell you a little bit about some of the things that we've been doing to understand better both how this targeting mechanism operates, and also how we can use that information to think about applying it for particular applications in human cells. So when people ask, why did this technology sort of take off as quickly as it really has, I like to point out that it's kind of like sort of software versus hardware. I think where the older, earlier technologies for genome engineering were hardwired, requiring new protein engineering for every experiment, here we have a system that's more analogous to software. We can reprogram a single protein with a guide RNA that's trivial to change the sequence of, and redirect it to new sites. And this, of course, has many applications in animals and plants. We'll talk about a few of those here momentarily. And as I'm going to show you, it's sort of amazing to discover that this is a bacterial system that's really evolved to have a very rapid and accurate DNA targeting mechanism. So it really makes it very useful for all sorts of applications, certainly for research purposes, but I think in the near future, probably also for clinical applications as well. So a couple other things about the system that have made it really fun to work with, and for labs adopting this for all kinds of applications have made it really something that can be applied for various kinds of experiments. First of all, this is a protein that is easily modified. So it's quite been trivial to turn it into a deactivated form of Cas9 or dCas9 that has the active sites mutated but retains the ability to bind DNA in a targeted fashion. So that it allows this to be utilized for things like transcriptional control and live cell imaging. It can be coupled to, this protein can be coupled to other types of functionalities. So people have been able to already show that you can use this to make epigenetic changes to the DNA, changes in methylation state and things like that. In bacteria, it's actually a protein that is naturally multiplexed. So in other words, in a bacterial cell that's using this as an immune system, this protein would be programmed with a variety of different guide RNAs to protect the cell from multiple different viruses at the same time, much like our own immune systems operate. And so that means that we can actually take advantage of that property in an experimental setting by programming Cas9 with multiple guide RNAs in a single cell for experimental purposes, doing things like making multiple changes to a genome in a single shot. And I want to say now a little bit more about the target recognition mechanism and what we've learned so far about how this little machine actually operates. And the first clue that it really is kind of a little machine was some work that we did with my colleague Eva Nogales at Berkeley using low-resolution electron microscopy. This was actually negative stain electron microscopy. Two students, David Taylor and Sam Sternberg, that did this work where they were able to visualize the Cas9 protein in different states of assembly, starting with the protein alone and then looking at its structure as it bound to guide RNA and then ultimately to a target DNA sequence. And we could already see, even at about 28 or 30 angstrom resolution, that the protein underwent a very dramatic change in its structure upon binding to guide RNA. Namely, the protein went from sort of a closed state to a state in which we could see very clearly a channel opening up in the protein where the DNA RNA hybrid ends up when this substrate is actually associated with the complex. So we now know a lot more about this conformational change, having looked at a whole series of crystallographic structures solved by Cas9, by our lab and several other labs. And so let me show you a movie that was made by a graduate student that sort of morphs between these different structures and really illustrates the dramatic nature of the structural change that occurs in the protein upon association with RNA and ultimately with DNA. Oops, sorry about that. Let's see. Here we go. So this will start off in, this shows you the protein alone structure morphing to the state bound to the guide RNA. You saw a very large conformational change in this part of the protein right here that creates the channel down the center that we could see by EM where the guide RNA strand actually ends up. And here, this is showing you the structure with the guide RNA and then morphing now to the structure bound to a DNA target strand. And you can see there's a further rearrangement of these domains of the protein to allow that hybridization to occur. And then this final change that you're seeing is actually an imagined, this is sort of just our imagination. We know this conformational change has to occur because this is actually one of the cleavage, the catalytic domains of the protein called HNH that in all of the published crystal structures is in the wrong place to actually cut the DNA. So we know there has to be this further conformational rearrangement that puts it in position to actually generate the correct DNA double-stranded break. And I won't show you this today, but we have a very new, very recent crystallographic structure that's been solved by two people in my lab, Fugo Zhang and David Taylor, that's now in press for publication that shows the structure of the Cas9 protein bound to an actual R loop. This is bound to an actual double-stranded, full double-stranded DNA target. And in that crystallographic structure, we can actually see this conformation of the active site. So we know this final change does occur when it's bound to a bona fide DNA target. So, so we sort of, that's just showing you kind of snapshots of the structure, but we also really want to understand the kinetics of this. How does this system actually search through an entire genome? How does it go through the whole human genome to find a single target site? And one clue to this came from some what was initially in vitro experiments done by Sam Sternberg and Cy Redding. This was a great collaboration with Eric Green's lab at Columbia, using a single molecule system called DNA curtains that his lab has established. And what we figured out from those experiments is that the Cas9 RNA complex has very rapid binding kinetics to DNA. So it binds very quickly to DNA and then releases it. So it's searching the genome not by processive sliding along DNA, but actually through rapid binding and release of DNA. And we found that when it interacts with one of these PAM sequences, a GG dinucleotide, that slows down the release of the complex, the off rate, such that there's time for interrogation of the adjacent DNA strands. And if there's complementarity with the guide RNA sequence, then the DNA begins to unwind. If the complementarity proceeds, then unwinding occurs further to eventually form this full R-loop structure with the bound DNA opened up locally at the region of complementarity to the RNA, and these active sites of the protein then swing into place and make the double-stranded break. So, but this was all from in vitro data, so we wanted to really think about how this might work in a real nucleus with chromatin. And very briefly, I just want to show you some work that we published recently. This is a collaboration with Bob Tejan's lab and the great folks at Janelia Farm Research Campus to use super resolution microscopy to visualize Cas9 as it searches through the genome in living cells. And so the idea really was to explore how Cas9 is able to navigate not only chromatin structure, but also handle large genomes and deal with eukaryotic nuclear organization. And I'll just show you two things that we learned from this. So you're looking here at images. These are 10 millisecond snapshots in a live cell nucleus that is expressing a fluorescently tagged form of Cas9. And what we saw immediately from these experiments was that when Cas9 has no guide RNA bound, so it's the apo protein alone, or is programmed with a nonsense guide that doesn't bind to any site in the genome, these particles of the protein move very, very quickly. The kinetics are very fast in the cell, very rapid diffusion, and we can plot log of diffusion coefficient and frequency of number of particles on the y-axis. And you can see that in both cases we get plots that look very, very similar. However, if we program Cas9 with a guide RNA that recognizes a large number of sites in the genome, one of these repetitive sign elements in this case, we now see that a large number of particles have significantly slower kinetics. And you can almost see sort of a biphasic behavior. Some particles still with very fast kinetics in the cell, but a large number that have slowed down. And from making measurements of these kinetics and looking at changes when we change this guide RNA to have mismatches to the target sequence, we found out that Cas9 really spends very little time on off-target sites. It's again, very rapidly releasing sites that have a close but not perfect match to the guide RNA. But when it gets onto a site that has a perfect match to the guide, then it tends to hang around for minutes, if not longer. Okay, and let me just show you one more experiment that we did using this system. So we wanted to understand how Cas9 deals with highly compacted parts of the genome, heterochromatin. And in this experiment, what we're looking at are, again, cell nuclei in which cells are expressing a fluorescently labeled form of HP1. It's heterochromatic protein 1. That's part of, sort of marks where heterochromatic regions are located in the nucleus. And we can see these tracks of Cas9 that are fluorescently labeled moving around the nucleus. Doesn't look like much when I show it to you like that, but if we stack up these images, you can actually see that these Cas9 pathways through the nucleus are occurring largely in euchromatic regions, but we do see forays into these highly compacted heterochromatic parts of the genome. And that's very interesting because it actually corresponds to what we see experimentally, where we can actually get targeting of sequences that are located in heterochromatin, but with slower kinetics. And so we think that somehow this protein is able to deal with these compacted regions, perhaps telling us something about both the behavior of this protein, but also perhaps the dynamics of chromatin structure. So finally, in just the last few minutes, I just want to come back to this issue of how do we think about this in the context of precision medicine and personalized medicine. And I think that, from my perspective, there really are three major challenges right now to therapeutic applications. And I don't have on here target specificity because I think that this protein is actually really highly specific. I think data that I've seen from our own experiments and results from Rudy Yanish and George Church and others, that when this protein is used in appropriate amounts in primary cells or animals, the level of off-targets that are detected are extremely low to undetectable. So it's really quite an accurate system. But I think the real challenges facing us right now are delivery. How do we get this into cells or tissues where we'd like to affect editing for therapeutic purposes? How do we control the way that DNA is repaired after it's cut by Cas9? And what are the ethical and societal implications that come up when we think about using this in various contexts? So let me very briefly tell you about work that we're doing in the lab. This is actually an expanding area of my lab right now to think about how we can deliver this system. And as biochemists, we started thinking, maybe we could just actually deliver this directly into cells or tissues as a pre-assembled protein RNA complex. And the idea here is really to avoid having to introduce foreign nucleic acids into cells, such as DNA or RNAs that encode the protein or guide RNA, but actually just pre-assemble the protein and one or more guide RNAs in vitro, and then figure out how to introduce these assembled RNA protein or RNP complexes directly into cells. This has some important advantages, I think, for applications. First of all, we can detect editing very quickly within just a few hours of introducing this into cells, so it's a system that operates very fast. The half-life of the protein RNA complex is quite short, so this again minimizes any potential off-targeting that might occur. And we can co-deliver DNA templates for repair, so it helps to enhance the rate at which you get incorporation of new DNA at the site of the break by homology-directed repair. And I won't go into the data here. Some of it's already been published, but we have a wonderful collaboration going on with our colleagues across the Bay, Jennifer Puck, who's here at the meeting, as well as Catherine Schumann and Alex Marson, her colleagues at UCSF, David Martin at the Children's Hospital of Oakland, and Jacob Korn in the Innovative Genomics Initiative at UC Berkeley and UC San Francisco, in which we've been able to use the RNP delivery to rewrite the sequence in primary human T cells. We hope to use this in the future, not only for research purposes, but also, we hope, for real clinical applications. And just in the last minute, I'm going to say a little bit about ethics. So I started asking myself a couple years ago, what should we do now that genomes can be edited quickly and really relatively easily? And it occurred to me that that this works in certainly works in mouse embryos, rat embryos, various other kinds of certainly people had started to. There was a paper that came out in early 2014 doing editing in monkey embryos and showing that you could make genetically modified monkeys using this system. And I started to think, gee, when will it be the case that someone does an experiment in human embryos? And so this really got me thinking that we needed to begin to pull together the scientific community, clinicians, and other stakeholders to discuss the ethics and societal considerations around this. And so we had an initial meeting in the Napa Valley just about a year ago now with just a small group of scientists, David Baltimore, Paul Berg, and others who had been involved in the Asilomar meeting in the 1970s around molecular cloning were there. And that led to publication of this paper, which was published last March in Science magazine, in which we proposed what we called a prudent path forward, really asking scientists to refrain from using this kind of genome editing for any kind of clinical application in human embryos until this kind of application could be discussed and the ethics and safety and other kinds of considerations thoroughly considered. And so the good thing about this, I think, was that it really triggered a lot of people around the world to start thinking seriously about this issue. And we had a meeting last month in Washington sponsored by the Chinese Academy of Sciences, the Royal Society in the UK, and the National Academies of Science in the US, in which we discussed these issues. And you may have seen that proceeding, but basically that has triggered set of additional meetings that are now happening this year, and I think you'll see a lot more discussion about this in the media as well. So stay tuned to that. And I want to just close by acknowledging a number of people. It's been a really exciting experience for me, going from being a RNA biochemist and structural biologist to thinking about the applications of a technology like this in various systems, and I couldn't do it alone. So we've had really a lot of fun working through the Innovative Genomics Initiative. It's a UC Berkeley, UCSF collaboration, these various folks involved, and I invite you to check us out on the web. This is going to be an expanding effort. We have been very fortunate to have some philanthropic donors, and we also have partnerships with various companies to try to think about and apply the genome editing technologies to particular disease targets. And finally, I couldn't do anything without a wonderful group in my laboratory. So I mentioned a number of these students and postdocs along the way. I also want to highlight our collaborators. I mentioned, I think, all of these folks that we've been involved with. Alex Marson, in particular, has been terrific for helping us get into the thinking about clinical applications of this. And finally, I'm really grateful to our funding sources. And I want to give a particular shout out to the National Science Foundation that has funded my work over the last several years and actually gave me the first grant to work on CRISPR biology back before there was any information or any really any interest from most groups around the world. So thanks to them, and thanks to all of you.